So, good afternoon. I'm Sveva Pelliccia, a postdoctoral researcher, member of the organizing committee of this virtual conference, and I will be also the chair of this session. So we are ready to start with the second medicinal chemistry session, introducing our keynote speaker. It's a pleasure for us to have with us Professor Angela Russell from University of Oxford. Professor, are you with us?
I am, yes, I'm just share my screen.
Okay, so I have summarized their great CV, and I hope I have not forgotten anything. Angela Russell gained her medicinal chemistry degree in 2000 and her PhD in organic chemistry in 2004. In 2007, she was awarded a Research Council's UK Fellowship in Medicinal Chemistry. In 2019, she became Professor of Medicinal Chemistry. Her research interests are centered around the discovery of new molecules and mechanisms to manipulate cell fate and to translate them into therapeutic agents, particularly for degenerative diseases. She won a lot of prizes during her career. In particular, in 2016, she was named as a rising star in the BioBit 50 Movers and Shakers in BioBusiness Report. And in 2020, she was awarded a Harrington UK Rare Disease Scholarship in recognition of her work on developing a therapy of Duchenne muscular dystrophy. So congratulations for your research and your prizes. And we look forward to hearing your presentation entitled "The Cures from Within. Can we use chemistry to teach the body to heal itself?" Please, Angela.
Okay, thank you for that very kind introduction. And thank you to Stefano and others on the organising committee for inviting me. It's a real pleasure to be able to come and talk to you about my work and a little bit about my career and how I've got to where I am. So I just want to start my talk by where we began and what one of the things that inspired us to get into this field, this idea of can we develop drugs that will stimulate repair and regeneration. So this was a quote from Ramon Higaral in 1928. So he'd won the Nobel Prize in Physiology several years earlier. And what he famously stated, and he was talking about the central nervous system, was that once development was ended, everything must die. Nothing may be regenerated. And of course, what he went on to prophesize was that it's for the science of the future to change, if possible, this harsh decree. And I think that's just a wonderful statement about what we might be able to do and the aspirations we might be able to realize. So I think if you ask most people what they think of as regenerative medicine, they would consider cell therapy. And that, of course, for the last 15 years or so has been one of the major things that has gone into the clinic in this area. And so the idea here is you take cells either from the patient themselves or from a donor. We grow them up outside of the body and then inject them directly into the patient to initiate repair and regeneration, or alternatively, graft onto a matrix, be that real or artificial, and then introduce that. But there's been huge challenges in this field, and as of today, there is still no medicine that is used as stem cell-based medicine that is used as a standard of care above other therapies. There's issues of cost, immune rejection, and standardization, and so on. But what was really intriguing to us as we were coming into this field is that this was a review that was published in 2012, summarizing essentially the first 10 years of cell therapy trials. And this was a figure buried in this paper, which talked about, well, what happens to the cells when they're injected? So even if we see a therapeutic benefit, what actually happens to the cells that have been introduced? And the results are quite astonishing. So what you can see here is that in 50% of cases, there's only a transient dosing of the cells. So these cells are being cleared in a matter of days or weeks following injection. So this really flies against the idea that this is a cell replacement therapy for regeneration, rather that there is some indirect mechanisms which actually are stimulating an innate repair and regeneration pathway. And that's what really intrigued us, because it really suggested an alternative way that we could think about stimulating these processes, whereby you could negate the need for transplanting cells at all by directly stimulating either the tissue resident stem or progenitor cell populations, or otherwise conditioning the environment to stimulate repair and regeneration directly. And this is an area that we've been in for some years. And we now know, and this is a lovely review, this is an image taken from a lovely review from James Wells and Fiona Watt a few years ago, reviewing what is now understood about mammalian regeneration and regenerative capacity. So we know now that many organs and tissues in the mammalian body, including humans, can regenerate themselves throughout life in normal homeostatic processes, but also, and most importantly, there is a response to injury and disease where we stimulate these regeneration and repair pathways. But the problem is, of course, is that endogenous capacity is usually insufficient if we have an acute injury or a chronic degenerative disease. And so what we and others in the field are interested in asking is, if we could stimulate those processes, these natural processes, might that be a viable therapeutic approach? And there are, of course, drugs. We've reviewed this area a few years ago. We've got another review that will be coming out in the next few months. But there are drugs that act via these sorts of pathways that are already in clinical use. Not that many, but there are a few. And so for us, we're in a number of areas where we're a medicinal chemistry group, but we're really interested in addressing these questions about how we can stimulate repair processes in the case of either degenerative diseases, or the flip side of that, which I won't talk about today, is in proliferative disorders, so oncology and so on. So I'm going to talk to you about two stories from my lab. One is really how we got into this field in the first place, which is our work on this rare disease, Duchenne muscular dystrophy, and then how those learnings and how the things that we discovered as a part of that inspired us to move into regenerative medicine more broadly. So I'll give you a little bit of background about Duchenne muscular dystrophy, because this is a rare disease and not everybody may have heard of it. So this is as I say, it's rare. It affects about 1 in 5,000 live male births. But actually, although it's rare, it's in fact one of the most common fatal genetic disorders, which is diagnosed in children. It's an incredibly debilitating, progressive, fatal muscle wasting disorder. It's caused by mutations affecting the dystrophin gene, which is a very large gene encoded on the X chromosome. And what happens, there's many different mutations, but the phenotype is similar. You cannot produce a functional form of the dystrophin protein. So this is showing you here an image of a section of muscle fibers from a healthy individual, and here's dystrophin at the membrane. And when you look at a corresponding section from a Duchenne muscular dystrophy patient, it's characterized by the complete absence of dystrophin at the membrane. It's very striking. And there is no cure. We have some treatments that have gone recently into the clinic, but they're not yet very efficacious. And there is a real need for treatments that will show real benefit or can cure this condition. So these are generally the ways that people are exploring in order to try and treat this disease, either by restoration of the missing dystrophin, and there have been some extraordinarily innovative approaches that have been taken, but they have their limitations. They're not relevant for all patients, particularly these exon skipping approaches, which target very specific mutations. Our approach was very different. It was to say, well, can we actually replace the missing dystrophin, but a related functional protein called Eutrophin? And the advantage that we conceived of in this approach is that in principle, it would target all of the patients, irrespective of their underlying genetic mutation. So Eutrophin was discovered by Professor Dame Kay Davies in Oxford, and with whom we've been collaborating for nearly 20 years. And this is just showing you a schematic of those two proteins side by side. And what you can see is that particularly in the N and C terminal regions, these are very similar to one another, more than 95% identity in the C and N termini. And they provide a similar link in the muscle, which is, so zooming in on a muscle fiber, they provide this critical link between the actin cytoskeleton, the contractile machinery within a muscle fiber, and the cell membrane. So this is a dystrophin-associated complex, and it's really responsible for the integrity of the muscle in exercise-induced contraction, and protects against injury of the muscle. So what we asked is, could we actually replace, because this link is missing in Duchenne muscular dystrophy patients, if we increase levels of Eutrophin, could we replace that missing link with an alternative functional protein? So the way that we went about doing that, and to say this work was initiated nearly 20 years ago now, was to develop a screen whereby we could look for molecules that acted on the promoter, so the region of DNA upstream of the Eutrophin gene, where we looked at production of a surrogate enzyme called luciferase, where we could then correlate the luminescent output from this process with the activity at the promoter. And we wanted to know what sort of molecule was going to work in this context. So we didn't know very much about the regulation of expression of Eutrophin. We knew that some of the players involved were nuclear receptors, particularly nuclear hormone receptors, of which the ligands are well known as sterols. So we thought it made sense to begin by looking at some of these still bean type motifs, because these are known to be structural mimics of those sterols, those modulators, those endogenous modulators of those nuclear receptors. So we made a few examples of these, and what we were really pleased to see is that even in that first set that we made, we found molecules such as this compound here, where you can see as we're increasing the dose on the x-axis, we're seeing an increase, a dose-dependent increase in luciferase activity, the luminescent output in this reaction. The problem with this, and this is to a medicinal chemistry audience, I'm sure you will appreciate already, is that these are not suitable molecules for progression into in vivo studies, although they're very useful in vitro tools, because they're subject to oxidation. And in our hands, we see that these epoxidize this double bond in the molecule. So we need to switch to an alternative bioisosteer, which is not susceptible to that oxidation. So we did that by switching to these benzoxazoles, amongst other heterocycles. And this molecule here is an example of one that we got quite excited about, because it both recapitulates the activity in the luciferase assay, but also when we looked at message RNA level in muscle cells, we could see an increase in the level of Eutrophin RNA and also protein, although I'm only showing you the RNA data here. So part of my career has been in the academic sector. I've also been involved in thinking about how we can translate our work into the clinic. So we've spun out a few companies over the years, and one of the early companies I was involved with was as a result of this work. So what happened is that the work I've described to you happened in the early 2000s. There's this generation of these benzoxazole Eutrophin modulators, and that led to the formation of a spin-out company, originally called Vastox. It's now called Summit, still is called Summit Therapeutics, that sought to commercialize and develop a drug out of these early stage findings in our labs. And over the course of the few years, they did just that and developed this drug called Zootremends, so directly inspired by the work in our lab. We carried on working on this and spun out a second company, which I'm not going to talk about today, and we formed an alliance with the University of Oxford to take forward this pipeline of Eutrophin modulators in 2013. I'm going to take you through the clinical data, but it's not a happy story. But what I hope I will leave you with is a more positive note, because we have been able to reactivate this program. And I think we are now much closer to being able to find a cure than we were before. I'll take you through that data. So this is a Zootremend, and I'm just going to share with you some of the data that convinced us that this was a good molecule to take into a clinical trial. So this is data from dosing a mouse model of the disease. This is the MDX mouse, 28 days, once per day, at 50 milligrams per kilogram. So what we first of all look at is upregulation of Eutrophin. So this is the untreated mouse here. So you can see there's not very much staining of Eutrophin at the membrane. It's a much more consistent staining across these muscle fibers when we add in a Zootremend. But most importantly, we can correlate that increase with a reduction in degeneration and regeneration of the muscle fibers. There's less muscle damage. That's true in both the diaphragm, which is shown here, but also in the soleus, which is a skeletal muscle. And then when we look at the strength in the muscles, so this is an assessment of forelimb strength in the mouse. This is what a wild type mouse looks like when you exercise its forelimbs in terms of the force that it can generate. When you exercise a mouse model of muscular dystrophy, you get this huge drop in the force it can generate. But if you exercise a mouse that's been, an MDX mouse that's been treated with a Zootremend, you restore that function to near wild type levels. So we thought this was really exciting, and this was what drove us to take this into the clinic. So this is, I'm going to share with you the data from the phase 2 clinical trial. Obviously, it went through some earlier clinical studies, which I won't discuss. So this study was an open label study, which is normal in a rare disease like Duchenne. We took 40 boys between 5 and 10 years old in the US and the UK. We monitored them at baseline, and then we treated them, measuring again at week 24, and then at 48 weeks, which was the end of the trial. And what we saw was really encouraging halfway through the trial. So one of the markers that we looked at was a protein called developmental myosin, and this is a marker of degeneration in the muscle. And what you can see, this is taken from one patient at baseline. So all these fibers here stained in blue are expressing developmental myosin. So they are newly regenerated in this muscle. And you can see there are an awful lot of them, which is typical of a patient with Duchenne. After 24 weeks of treatment, you can see by eye that there are very many fewer of these newly regenerated fibers. That's just one patient, but when we looked across the cohort, we could see the statistically significant and meaningful decrease of 23% in expression of this regeneration marker across the cohort. And that correlated with an increase in Eutrophin and a decrease in inflammation through other measures. So this looked incredibly encouraging. But when we looked again at the 48 weeks at the end of this trial, it was as though these children had not been on the drug. The disease had followed the natural history. And so what we really needed to, is obviously incredibly disappointing, not just for us, but for the patients and their families. And it really made us want to understand, why was there no sustained clinical benefit? Why did we see this positive effect 6 months in, and then the effects went away? And so over the last 2 years, we've done an awful lot to try and understand why that was the case. And I think we do understand now why that happened. So the first thing to discuss is Zootremends metabolism. So a Zootremend structure is shown here. We already knew also undergoes a cytochrome P450 mediated oxidation, mediated by the CYP1A2 predominantly, to these 2 regioisomeric dihydrodiols, both of which are inactive as Eutrophin modulators. But what we also observed, and this was in the early phase clinical trials, that when we look at the exposure from day 1 through to day 7 or day 10 and then on in the trial, we see this reduction in peak plasma levels in the area under the curve. So this is showing you, this top curve here is the exposure in healthy volunteers on day 1, and then these 2 curves at the bottom are day 7 and day 10. So there's about a 20% reduction in the area under the curves, the exposure of this drug. And that's because this drug induces its own metabolism. So we induce an increased levels of CYP1A, which we subsequently showed, and that's what's leading to this increased metabolism and this reduction in exposure. So this is a problem, but when we looked at this, it's stable after 10 days. And we thought, well, if this should be still enough, that we get enough drug in when we go into the trial with the patients. But what we were not expecting is that when we went into Duchenne boys, they were much more prone to induction of metabolism. So the reduction was not 20%, 60%. And simply, we think that a large portion of this is because at the beginning of the trial, we have enough drug on board to elevate Eutrophin. Eutrophin is actually surprisingly stable at the muscle membrane. The half-life has been measured by others. It's about 4 to 6 weeks. And so gradually you are losing this expression from the membrane, and that's why you can still see effects, positive benefit out quite a long way into the trial, but obviously that effect cannot be sustained. So that's what we believe is going on as a reasonable hypothesis based on the data that we have. So what we needed to do was really understand how Zootremends was working and whether we might be able to develop a drug with a common mechanism that could evade this induction of metabolism. And so we use, and this is routinely what we do in my lab, a variety of techniques to try and understand that problem. So these are, I'm not going to explain all of them, but they're shown some of the representative techniques at the top, which we use together. I'm a firm believer that we need multiple assessments to try and come and then draw a common conclusion from these. It's the best and most efficient way in which we can drill down into molecular target and molecular mechanism. And if you're interested, we've reviewed this. A paper came out last year. But the mainstay of the technologies that we use is chemoproteomics. And so here we design a probe, and it's a technique that many people may have come across already, but for those that may not, you design your probe such that it has a photoactivatable group at one end, and then a clickable handle at the other. So we can mix this with the muscle cells. We irradiate then at a wavelength which is not damaging to biological molecules. That activates this photoactive group. We form a covalent bond to the target of interest. We can then lyse the cells, conjugate to either biotin or a fluorophore, which allows us to then analyze the proteins or other biomolecules to which this is bound, either through in-gel fluorescence or through proteomics from that enrichment process. So we knew enough about the SAR of Zootremends that we could reasonably design these probes where this photo affinity group mimics the sulfone. And then this alkyne, which we use as the click handle, if we introduce this either meta or para, it can recapitulate the phenotype, as I'll show you in a moment, of Zootremends. We also knew that if we change the regiochemistry of this benzoxazole from the 2 position to the 1 position, we completely lose the ability to upregulate eutrophin. So we use that molecule as a negative control in our experiments. So I'm not going to take you through all of the validation data. This is work that was published last year in this paper down here. I'm going to take you straight through to the conclusions. So what we did, so this is just showing you the in-gel fluorescence where we could see some changes between the two, but really the informative data came from the proteomics, the chemoproteomics experiment that we did. So this is just showing you a typical volcano plot where any of these proteins up in this region are statistically significantly differentially enriched in our affinity probe treated samples. And when we, so this is just showing you DMSO, the vehicle control versus the affinity probe. When we compare a treatment with a competitor molecule or with a negative control, we can see that there are a set of proteins up here. And the one we were most interested in was, for various reasons, was the aryl hydrocarbon receptor, although we did look at the others. And so we went on to show several lines of evidence that it does indeed bind to the aryl hydrocarbon receptor in vitro. So this is showing you data from a purified recombinant protein. And also in live muscle cells through Western blotting, we were able to show this affinity probe binds to aryl hydrocarbon receptor, which is shown here. The negative control does not, and we can outcompete the reaction of the affinity probe by using either a Zootremends or another related competitor molecule. And functionally, we're able to show that this acts as an antagonist of the aryl hydrocarbon receptor in muscle cells. So this is just showing you what happens when an agonist binds. You get translocation of AHR to the nucleus, where it dimerizes with ARNT to induce transcription downstream. And that's what you're being shown here. This is in the presence of an AHR agonist. You can see this green stain for AHR, where AHR is both in the nucleus and in the cytoplasm. If you treat with an antagonist, what should happen is you block nuclear translocation. And when we look with a Zootremends, we can see that's exactly what happens. Okay, so the blue dots here are the nuclei, but you can see there's no AHR now in the nucleus, probably clearest from this merged image. And when we look then downstream at the target genes that are transcriptionally regulated by AHR, we can see in both MDX, which is the mouse, and DMD, which is the human, we get this reduction in transcription of AHR-dependent genes. So that's all well and good, but is an AHR antagonist going to always be a eutrophin modulator? So to test that, we took, and we've now tested 12 completely structurally distinct, bind in different ways to the ARL hydrocarbon receptor, and all of them in human cells and mouse cells show an increase in eutrophin protein. This is showing you data from a few in human dystrophic muscle cells. And you can see across the board with these blue lines here, we get this increase in eutrophin expression. So we genuinely think that AHR it's the target of Zootremends, and it's the target that's leading to the elevation of eutrophin. So we are, and I'm going to talk that much more about that, pursuing this in our labs at the moment for the next generation compounds to take to the clinic, which can circumvent the issues of Zootremends. So what I just want to finish on in the last few minutes that I have talking to you is how this work has inspired us into other areas. And one of the key observations that we made is that actually eutrophin is relevant not only in developing muscle, which is how it was originally discovered, but also in repairing muscle. So this is a cartoon showing you a muscle fiber. And what you have dotted along the side are these satellite cells. These are the muscle stem cells that all of us have in our muscle. And when the fiber is damaged, what happens is that these satellite cells, which are expressing eutrophin, will migrate into the area of injury. They will proliferate, they will fuse to form a new myotube. And when the newly repaired fiber is generated, it has eutrophin and not dystrophin at the membrane. So what this told us is that we reuse these fetal or development programs in regeneration. And if this paradigm was relevant in muscle, is it used across all sorts of other tissues? And the answer to that question is very definitely yes, and others have shown that too. And so the second question that then arose from that is, if this is true, can we stimulate these types of processes using small molecules to stimulate repair and regeneration? And this was really one of the key findings for us to inspire us to go out into all sorts of other areas, including looking at cardiac regeneration, neuronal regeneration, pancreatic regeneration, and so on, all using a similar paradigm. So I'm going to very briefly just touch on the work that we've done in neuronal regeneration. I realize that I have to stop to take questions, but just to tell you that we've looked at this now, looking at both neurogenic niches. You have two niches in your brain that can make new neurons throughout life. We set up a screen in order to monitor this. And we found, and this is just showing you the molecule that we found from that screen in vitro in both regions. This is the subventricular zone on this side and the dentate gyrus on this side. And what you're looking at in pink are the new neurons. And you get this really quite dramatic dose-dependent increase. And we've taken that on. It's orally bioavailable. And just to finish for you, to say you can translate these findings also in vivo. So this is showing you the results of an experiment where we dose that same compound that I've showed you working in vitro in the mouse, and we get this stimulation, marked stimulation of neurogenesis in the mouse brain, both for new immature neurons, but also these neurons mature, survive, and integrate. And so that looks incredibly exciting. We're really, really excited by this data, especially because it correlates with an improvement in cognition and function. And so I think there's some really important and exciting future work to be done in here, and seeing how this can be applied in neurodegenerative diseases or diseases associated with the cognitive deficit. So I'm going to finish where I started with Cahal and pose a question, because can we develop effective therapies that will stimulate these repair processes? I think the answer to that question is definitely yes. It's going to take some time, but I think it's an incredibly exciting and vibrant area, and we shall see what comes in the next few decades. And with that, I'd just like to thank everybody. This is a pre-COVID photograph, and we're all looking very glamorous in my group here. Particularly, I'd like to point out Isabel Wilkinson here in the middle, who described single-handedly did all of those that target deconvolution work of Zootremends that I showed you in the middle. But thank you. This is work with a lot of collaborators, a lot of interaction with funders and charities, and I'm very grateful to all of those. And thank you very much for your attention.
Thank you, Angela, for your brilliant presentation and for this journey around the neurodegenerative diseases. And okay, any question from the audience?
I have one. Hi, Angela. I'm Stefano Tomassi. Congratulations for your presentation. I have actually one curiosity and one question. So the curiosity is if this new paradigm can be applied to other diseases, and the other one what is the solubility of the Zootremends, the compound that you show us, and how did you, in the end, if you try to cope with the metabolic instability of that naphthyl ring?
So the first question is probably the easiest one to answer because it's yes. And so, and there are a lot of, we are not alone, of course, in this field. There are lots of other people looking at regenerating other organs using similar approaches. And it does look like the most successful approaches that I've observed are those where we try and enhance natural processes that are already there to try and stimulate repair and regeneration response to injury. They seem to be the ones that are working best so far, but we're still away from seeing those being applied and showing success in the clinic. Your second question was around Zootremends and the solubility, and you're absolutely right, it's awful. And in fact, the reason that it took so long to get it from the discovery into even a phase 2 clinical trial was because it needed to be carefully formulated. So we got around the challenge of exposure in terms of absorption, but then you're left with this problem of metabolism. And you're right, so we've published some work which I didn't have a chance to talk about today, where we can replace both the sulfone, which is at least in part responsible for its poor solubility, and the naphthol, which is the major point of metabolism. You need something relatively lipophilic in that region, it turns out, which makes sense. Now we know it's the aryl hydrocarbon receptor, so particularly fluoro and trifluoromethyl aromatics seem to be a stable replacement that we can use, but there are some other things that we've trialed as well.
Thank you.
Okay, we have probably another question from Margherita Brindisi, a member of our scientific committee. She said, very inspiring talk, Professor Russell. I would like to ask you to probe your opinion on the importance in muscular dystrophy and other diseases of the availability of an animal model fully recapitulating human disease features.
Yeah, fully recapitulating, I think, is the key.
Yes.
Yeah. I think it's an excellent question, and it is a common stumbling block in any aspect of drug discovery. So for Duchenne muscular dystrophy in particular, so the mouse model only is a model of about 20% of Duchenne muscular dystrophy. It has a premature stop codon, which is similar to 20% of DMD patients. It's a much milder phenotype than the disease, but there are other models, mouse models that have been generated since, which are more closely follow the more severe disease progression that you get in humans. So that's one aspect, is about disease severity. And interestingly, probably, so it's a slight aside, but the mouse makes more eutrophin than we do in humans. And it may be that actually that protective eutrophin is the reason that the mouse doesn't get as ill with this disease. The other issue, and this is particularly relevant for muscle diseases, and it's a criticism that's been leveled at us in the past, which is completely fair, is that the mouse muscle is very much smaller. So if we're talking about regeneration of muscle, or protection of muscle from damage, it's much easier, in principle, it's much easier to protect something where there's less muscle mass than a human where there's way more. But actually, there are dog models of the disease. So the eutrophin modulators have been taken into, or eutrophin modulation has been used and shown to be effective also in a dog model, which is a bit more similar to the human disease. But yes, there are real limitations for all of these, and there's no substitute to being able to go into human patients, and the real test is always in human subjects. And I could go on for a long time about models of neurodegenerative disease. I'd probably better not in the interest of time, but they are severely limited. And I think the best way we can look at those is that they only recapitulate features of human disease. And that's probably how they're best used.
Okay. Thank you again, Professor Russell, and hope to see you next year, probably in Naples.
Yeah, hopefully.
And okay, we can skip into the oral communications. The first speaker is Ottavia Bellotto from University of Trieste.
Hi. Okay, now I'm sharing.
Ottavia, are you with us?
Yes.
Okay, you can share your screen.
Yes.
Okay, she will show us supramolecular hydrogels from uncapped short dipeptides, heterochirality, and halogenation as tools to inspire self-assembly. Please, Ottavia, the stage is yours.
Okay, thank you very much. Thank you for the presentation. So I am Ottavia Bellotto from University of Trieste in the group of the Professor Silvia Marchesan. And today I'm going to present my research work. Just a moment, there's a slide. Okay. So molecular self-assembly is a process in which single molecules tend to interact to form organized supramolecular structures, basically driven by non-covalent interaction, such as van der Waals force, hydrogen bonding, pi-pi stacking interaction that usually require low energy. The self-assembly is a process that is ubiquitous in nature and can occur at all scales. And we have several examples, especially in the kingdom of peptides. For instance, we can think about the ribosome, or we can think about, for instance, the spider silk, in which the spiders can use the silk fibrin protein to create the three-dimensional structure. Furthermore, peptides, thanks to their capacity of self-assembly and their fine organization, can also give to material of life useful properties such as tautness in the features, dynamism in the cell cytoskeleton, or they can be useful scaffolds such as the collagen in the cornea. Little by little, in research, peptides have gained a widespread popularity thanks to great chemical diversity, their biocompatibility and biodegradability, and the possibility that they offer to work in a green chemistry context. Among them, especially short peptides consisting of 2 or 3 amino acids are very attractive because of their simplicity to produce them with a low cost and usually high yield. So the several advantages that especially short peptides offer and their propensity of self-assembly can inspire the development of cost-effective bio-nanomaterials very useful in different biomedicine fields. But which are the forces that can drive this self-assembly process? In literature, it is reported that typically some rigid and aromatic moieties, such as the naphthalene group and SMOC group, besides features like aromaticity and hydrophobicity, are able to drive the self-assembly of these short hydrophobic dipeptides. We can think about, for instance, the single amino acid phenylalanine that is well known for its propensity toward the self-assembly due to its aromatic and hydrophobic features. But we can also think about the diphenylalanine in the case of the dipeptide that is well known in literature to be a minimalistic self-assembly motif and also that can form a versatile array of different supramolecular structures. However, this dipeptide has some drawbacks, such as uncontrolled hierarchical assembly and well-known cytotoxicity. So the question, our question is, how can we overcome these two drawbacks? In a recent published work of our group, we demonstrated that chirality could be an effective strategy to overcome these two drawbacks and to generate hydrogel biomaterials and control the self-assembly of such unprotected dipeptides. In this work, XRD data reveals that despite the different stereo configuration of these two peptides, both are able to stack into nanotubes. The different chirality of the N-terminal dictated the direction along the tube, left-handed for the homochiral peptide and right-handed for the heterochiral peptide. The orientation of the aromatic chains between the two stereoisomers is different in the space, and so we can observe face-to-face intramolecular interaction only for the heterochiral dipeptide. This inner nanotube cavity is described such as an amphipathic water channel with an hydrophobic layer outside and hydrophilic cavity inside. In both stereoisomers, the hydrophobic interaction can lead to a double tube layer of peptide. As we can see, in black is the first peptide layer, in gray and in blue light is the second tubular layer of peptide. However, because of the different orientation in space of the aromatic side chains between the two stereoisomers, only for the homochiral peptide we have a hierarchical assembly in two bundles due to this rugged outer surface and the interdigitation between aromatic rings. By contrast, no hierarchical assembly occurs for the heterochiral peptide, where there is a smooth outer surface, and so the peptide tends to organize into fibrils with an homogeneous diameter of 4 nanometers. As we can see from 10 micrograph, we can observe the rigid microtubes and these homogeneous fibrils for the heterochiral peptide, so the different supramolecular organization. So the result is that the heterochirality can be an effective strategy to get hydrogels biomaterials different each other. And for instance, for the heterochiral peptide, we can get the transparent hydrogel stable over time, thermoreversible, and with a good cytocompatibility versus mammalian cells. So after seeing the versatility of the diphenylalanine motif and the chirality that play a key role to get hydrogens biomaterials with a good biocompatibility, the question is what's happened if we introduce an electronegative atom such as an halogen in one of these two aromatic rings of the heterochiral dipeptide. It's not so easy to predict a priori because both steric and electronic factors play a role in this process. So to this end, 4 of these compounds were synthesized, and they differ for the presence of the fluorine or the iodine and the position of the fluorine on the aromatic ring. The halogenation results also this time like strategies to tailor the supramolecular packing of these unprotected dipeptides. In particular, for the fluorination, we can observe an analogous packing that remember the supramolecular packing of the diphenylalanine dipeptide non-halogenated. In fact, the presence of the fluorine preserved this nanotube formation with an amphipathic water channel inside. Furthermore, the fluorination, depending also on the position of the fluorine on the aromatic ring, can involve different inter-hydrophobic interaction between the peptide molecule. It can lead to a different bundles of fibrils, for instance, with a diameter of 4 nanometer, 12 nanometer, or 27 nanometer. By contrast, in the presence of the iodine, we don't have nanotubes and these water channels amphipathic anymore, but we can get layers of stacks of peptide with an alternation of hydrophobic and hydrophilic layers, probably due to an high steric hindrance of this atom, the iodine, that can't enter inside the nanotube. So at macroscopic level, the net result is the halogenation can get hydrogenous biomaterials with a different stability over time. In particular, in the case of the iodine, the stability is increased. However, we have in all these cases a low biocompatibility that limits the use such as scaffold for futures biomaterials. So in the end, this work focused on the use of unprotected peptides such as building blocks with low cost and good biodegradability to generate hydrogenous biomaterial under physiological conditions by using heterochirality and halogenation such as effective strategies to control and to tailor their supramolecular packing. The chirality results such as a good strategies to get stable, thermoreversible, and hydrogen with a good biocompatibility, especially in the case of the heterochiral dipeptide. Also, halogenation could be an effective strategy to get other biomaterials, but with a low biocompatibility that limits the use, such as a scaffold for future application. So, in the end, the most promising component for now is the heterochiral dipeptide phenylalanine, and future plans aim to include a bioactive motif to direct cell fates and to prepare advanced low-cost biomaterials. I would like to thank you for your kind attention. I would like to thank you all the collaborators and all my group for the support.
Thank you, Ottavia, for your nice presentation. And any question from the audience? Yes, we have a question from Stefano Tomassi, Dr. Stefano Tomassi. Congratulations, he has a curiosity. Have talked to other aromatic and not natural amino acids with larger aromatic system to study the assembly to naphthalene or one naphthalene, alanine?
At the moment, no. We are using other hydrophobic amino acids, such as, for instance, leucine or isoleucine. And our aim is also to work in aqueous systems. So it could be an interesting observation. Maybe we can, in the future, we could try, yeah.
Okay. If you don't have any question, thank you again.
Okay, thank you. Thank you.
Thanks. And it's the turn, now it's the turn of Sebastiano Intagliata from University of Catania. So, with a presentation entitled New 5-fluorouracil and heme oxygenase 1 inhibitor, mutual prodrugs with in vitro anti-cancer activity towards colon cancer cells. Whenever you want.
Yeah, good afternoon. I cannot share my screen because right now the other...
Probably because, yes, Ottavia, you have to close your screen.
Sorry, yes. Oh,
yes,
sorry.
Thank you.
Okay.
Thank you. So now we see your presentation.
Can you see my slides?
Yes, clearly.
Okay. Okay, good afternoon, everyone, and thank you to the organizer for providing me this opportunity. Today, I'm going to describe the development of 5-FU-based mutual prodrugs with an in vitro anti-cancer activity towards colon cancer cells. Colorectal cancer is the second leading cause of the all-related cancer death. And according to a WHO annual report, the estimated incidence of the of the colorectal cancer worldwide was about 20% in 2020. And in general, the life risk time of developing colorectal is about 4.3%, while the survival rates are largely dependent on what stage at which the cancer is caught. And among the FDA-approved drugs, the 5-fluorouracil is frequently used into the clinical as a standalone or as an adjuvant medication. 5-fluorouracil is a well-known antimetabolite which exerts its anti-cytotoxicity effect by follow the biotransformation into the active metabolites, following by the inhibition of the thymidylate synthetase or the incorporation of these metabolites inside the nucleic acid, both the DNA and the RNA. However, the clinical use of the 5-fluorouracil is limited by the undesirable clearance, the low route of the administration, and also some drug resistance phenomenon. So for this reason, the developing of 5-fluorouracil-based mutual prodrugs has been proved to be a promising medchem strategies to both improve the therapeutic effect of 5-fluorouracil or either to delivery, to target delivery into the cancer cells, the drugs. AMYC-oxygenase is a microsomal heat shock proteins, and the AMYC-oxygenase 1 or HO1 is the inducible isoform, which is mainly overexpressed during stress-related stimuli such as hypoxia, UV radiation, and also chemotherapy. At the cellular level, the AMYC-oxygenase is mainly located at the endoplasmatic reticulum, where it catalyzes the first step of the image degradation into the biliverdin, carbon monoxide, and also ferrous ion. All of these catabolites can exert the anti-apoptotic effect mediated by the activation of the HO1 and can promote the cellular homeostasis and the cell survival under stress condition. In addition, the HO1 has been detected in subcellular levels such as mitochondria and also in the nucleus, where it can promote the upregulation of downregulation of transcriptional factors involved also with the oxidative stress. In addition, the HO1 overexpression has been proved to be expressed inside the cancer cells, where it can promote the cell proliferation, the cell survival, angiogenesis, and also can lead to drug resistance phenomenon. So for all these reasons, we were wondering if the coupling of HO1 inhibitors can be useful with the 5-fluorouracil, can be a useful strategies to overcoming some of the liabilities of use of the 5-fluorouracil. So we developed these new mutual prodrugs where the 5-fluorouracil moiety has been conjugated with our previously reported potent HO1 inhibitors through a succinyl group, which act as a cleavable linker. So here I report the synthetic scheme for the synthesis of the new 3 hybrids. We initially synthesized, following pathway number 1, the 3 different HO1 imidazole-based inhibitors. Following these synthetic procedures, which is already published. And these compounds, inhibitor 9, 28, and 42, were then used in the last step of the reaction with the key intermediate 5-fluorosuccinyl, which was prepared in a large amount by the reaction of the 5-fluorouracil with formaldehyde solution, followed by the treatment with succinic anhydride in the presence of DMAP. Finally, the new hybrids were obtained by the esterification of this key intermediate with the corresponding aryl ethanol imidazole derivatives using EDC hydrochloride as a carboxylic acid activator. As I already mentioned, in order to exert the biological effect, both the parent compound, the 5-FU, and the inhibitors needs to be released from the hybrids molecule. So here I propose a degradation pathway, and we have hypothesized that esterase in blood are able to cleavage these, to break these bonds, esterase bonds. So we assess in vitro metabolic stability, in vitro using porcine esterase solution and PBS, which served as a negative control. And as we can see from this graph, all the three hybrids shows pseudo-first order kinetic hydrolysis rate, which is also which is suitable for the release of the 5-FU moiety and the other counterpart, and also with about 50% of the compound remaining up to 6 hours. We also test the inhibitory potency of the new three hybrids towards the in vitro activity for the HO1 using microsomal fraction of the rat spleen. And as we can see, despite we obtain a lower a lower inhibitory potency for the three hybrids, compound EB20 and EB22 keep maintaining a reasonable inhibitory potency. And in particular, this potency was also confirmed in intact cells, in the case of compound EB22, suggesting that the hybrid molecules is able to cross the cellular membrane. As a preliminary cytotoxicity evaluation, we select 3 different colorectal cancer cells, and we test 1 doses of each compound. And as we can see, among the 3 cancer cells, the HCT-W16 cells results in the most sensitive one, with compound EB22 was more potent even than the parent compound 5-FU. We then decide to further evaluate in biological assay this compound by producing a more comprehensive MTT assay, increasing the tested dose. And as we can see from the chart bar, the bar chart, here the inhibitors shows from low to moderate effect on cell viability, while the new hybrids shows a dose-dependent effects on cell viability comparable to that or even higher than that of 5-fluorouracil. On the other hand, compound EB2 results in the most potent of the series, and also the co-administration further support the synergistic activity of the new hybrids. Then we perform morphological analysis, and as we can see from the pictures, inhibitor did not show a significant reduction on cell density and morphology, while the compound, the hybrid 22, shows reduction in both the morphology without changing in the density, without changing the morphology. On the other hand, the co-administration markedly decreased both the morphology and the density of the cells. We then evaluate the possible involvement of the antioxidant pathway by measuring the overexpression of NRF2, which is a well-known transcriptional factor which regulates the expression levels of different antioxidant genes, including the HO1. So as we can see from the immunoblot and densitometry analysis, both EB22 and combo administration highly increase of about 5-fold the expression levels of NRF2, which is also consistent with the expression levels of the HO1, and in agreement with the ROS production inside the cell, which has been detected in the early phase of the administration, and which was more predominant for EB22 and the combo administration. Finally, we also evaluate the mechanism of death, which involved the apoptosis, by measuring the decreasing level of the pro-caspase 3 as a deactivation of the caspase 3 dependent pathway, And as we can see here, we have a clear decrease in the procaspase 3 levels, which is indicative of the involvement of the apoptotic cell death. Furthermore, the cell cycle phase distribution in cancer cells shows that EB222 highly increased the population in the sub-G OG1 cell cycles, which is indicative of the fraction of the apoptotic cells. So here we show the preliminary result concerning our 5-FU-HO1 mutant prodrugs, which serve as initial proof of concept for the validation of these new strategies to treat colorectal cancers. However, further in vitro and vivo study are needed for further evaluate the clinical significance of these new strategies. And finally, I would like to thank all the collaborators and colleagues at the University of Catania and the funding support, and you all for the kind attention.
Okay, great presentation, Sebastiano. Any question from the audience? We have a question. Stefano Tomassi. He said that he saw that the heme oxygenase 1 binding portion presents a stereocenter, not defined in the structure. So did you characterize the specific enantiomer or the racemic mixture only?
Yes, thank you for this question. Actually, in our previous work, when we developed these new HO1 inhibitors, we also perform a specific analytical methods for the to resolve the enantiomeric mixtures, and we test the different potency of the two enantiomers towards the HO1 inhibition. However, the pure enantiomers did not affect that much the inhibitory potency of the enzyme. So we then decide to further continue our manipulation by using racemic mixtures.
Okay. We have another question from Dr. Coluccia, University of Sapienza, our member of the scientific committee.
Hi, Sebastiano. Thanks for your presentation. Just a curiosity. Have you tested the compound, the single inhibitor with the linker? So the 5-fluorouracil derivatives with the linker. To
so
see
you
if the link.
You mean the inhibitor with the succinyl linker?
Yeah. To see how the linker affects the biological activity.
Okay, so thank you for your question. Actually, we did, and the potency of the towards the HO1 was actually affected of about 50-fold degree. So means that the potency is highly reduced. And this may be also the reason we are working on this because we are trying to evaluating different kind of spacer because
it was my second question.
Yeah, I got your point.
So
based on the pathway that we propose, most likely the first degradation, the release of 5-fluorouracil is faster than the other ester bonds. So we are now tuning this hydrolysis rate in order to achieve at the same level, the same range of concentration for both the counterpart.
Okay, thank you.
You're welcome.
This was my question. Also, I will ask you if you consider other cleavable groups in the linker space.
Okay.
So, photocleavable or other
okay.
groups.
Yeah, okay. We did work on photoactivating photodonor in other series, but we didn't think about applying this kind of cleavable linker in these kind of series and prodrugs, but for sure this will be a good suggestion. So I will take note about this suggestion, and we may further develop this kind of photoactivating prodrugs.
Okay. Last question from Simona Musella. She has a curiosity. have you tried to evaluate the effect even in a more complex cellular system, like a three-dimensional organoid system?
Not yet, because actually here I just present a very small part of the biological evaluation. So we did we did test a lot of autophagical and other markers for the apoptosis, but for the sake of time, we did not show all these results. And we didn't consider to test on 3D cell models, but for sure this will be another step forward our research.
Okay, thank you again, Sebastiano.
Thank you.
And we can skip to another speaker. That is Alessandra Benassi from University of Pavia.
Good afternoon.
Yes, good afternoon. With the presentation, photoactivatable B-shaped bifunctional quinone method precursors is a new class of selective G-quadruplex alkylating agents. So, Alessandra, the stage is yours.
Okay, thank you. Good afternoon to everyone. Today, I'm going to present you the last work of our laboratory about the development of innovative G-quadruplex alkylating agents. Before going into details of the experimental work, I will do a brief introduction about G-quadruplexes. As we know, DNA can adopt different types of secondary structures, for example, triplex or double-stranded hairpins. However, among them, G-quadruplexes represent a very important biological target. G-quadruplexes can be fold when we have a nucleic acid sequence reaching guanines. This one can interact and form this type of secondary structures characterized by two or more guanine tetrads stacked one on another, held together by hydrogen bond interaction and pi-pi stacking interactions. G-quadruplexes can be found in very important regions of human genome. For example, a lot of quadruplex forming sequences can be found in gene promoter regions, and they can regulate the gene expression. We can find G-quadruplexes also on telomeres, the DNA sequence responsible of protecting the genetic material. And in the end, we can even find G-quadruplexes on untranslated regions of RNA sequences, and in this case, they are involved in gene translation. So G-quadruplexes have a very important biological role. So they represent an interesting biological target to develop therapies against different types of pathologies, including cancer. For example, quadruplex forming sequences on promoter region of oncogenes represent an important target to develop innovative therapies against cancer. However, G-quadruplexes are not stable in physiological environment, and we need some tools to regulate their formation. For example, we can use small molecules able to interact with quadruplex structures to induce their formation and stabilize these structures in physiological environment. To date, several molecules have been reported as G-quadruplex ligands. Here I reported the most common and the most famous ligands. And as you can see, all of them are characterized by an extended aromatic surface, which is essential for the interaction with guanine tetrads through pi-pi stacking interactions. However, most of ligands reported to date suffer of a major drawback. They present low selectivity for the target. So the aim of our work was to identify alternative targeting strategies to target G-quadruplexes in a physiological context. So to do this, we decided to combine two different approaches. The first one was based on the use of a photoactivable process, in particular of a photoactivable ligand. The use of photoresponsive molecules represent a great advantage because light ensures an elevated spatiotemporal control of the process. And we decided to combine this strategy with the use of ligands able to form a stable covalent bond with the target after the photoactivation. In this way, the formation of a covalent bond can increase the stabilization of the target. So to do this, we designed six novel V-shaped ligands, as you can see here, characterized by an extended aromatic moiety and by the presence of these two phenyl aromatic group with a leaving group, which could be a dimethylamino moiety or its corresponding ammonium salt. Irradiation of this particular phenyl derivative can generate this type of intermediate, a chinomethide, which is an highly electrophilic species which can react with a minor group of nucleobases to generate this type of covalent adduct, as you can see here. However, in order to reach this purpose, we needed to face two main issues. We had to demonstrate that our ligands were able to efficiently generate the intermediate chinomethide. And in a second moment, we had to evaluate the affinity of these ligands for G-quadruplex structures and their ability to form a covalent bond with the target after photoirradiation. So we started with a synthesis of compounds, and for compounds 1, 2, 3, and 4, we started from this substrate. We performed two Sonogashira cross-coupling, and we obtained the ligand 1 and 3, which differs for the substitution pattern present on this phenyl ring. With a simple methylation reaction, we obtained the ligands 2 and 4, which are the corresponding ammonium salts. The synthesis of ligands 5 and 6 was a little bit longer, and we started from this compound. We performed a coupling reaction to generate the corresponding amide. We performed the reduction and two Sonogashira cross-coupling, and we obtained the final naphtho-base ligand 5. With a methylation reaction, we generated its corresponding ammonium salt 6. At this point, we had to evaluate the photoreactivity of these compounds, and we had to demonstrate the formation of the expected chinomethide. Upon photoactivation, this type of phenyl moiety can generate the chinomethide, and its formation can be demonstrated by observing the formation of this hydration product. So we have performed irradiation of our ligands in water solution, and for each compound we have measured the photochemical quantum yield, which is a parameter that measures the efficiency of this photochemical process in this case. In this table, I reported the obtained results, and as you can see, all compounds showed good reactivity, but in particular, compounds 2, 4, and 6 showed higher photoreactivity. These compounds were characterized by the presence of ammonium salts, and the higher reactivity can be explained by the fact that the ammonium salts is a better leaving group compared to the dimethylamino moiety. Each reaction has been monitored through HPLC and Lcms analysis to demonstrate the formation of the expected hydrated product. Here I reported the case of compound 4. After 10 minutes of irradiation, we observed the formation of the expected hydrated chinomethide. And in a less extent, we observed also the formation of a bis-hydrated product. This is particularly interesting because it means that these compounds can act also as cross-linking agents. We observed the same reaction pathway for all ligands except for the naphtho derivative 5 and 6 that didn't afford the expected hydrated products. So at this point, we started with the evaluation of the affinity of these ligands for the biological target, and we focus our attention on two particular G-quadruplex structures. the telomeric quadruplex reported here, and the quadruplex present on the promoter region of the oncogene c-MYC. Both targets are important because they are cancer-related, but they differ for the type of structure. We have evaluated the affinity of the ligands through threat melting analysis. So we have measured the increase of the unfolding temperature of the oligonucleotide induced by our ligands. As you can see, all compounds show basically similar affinity for both structures. We performed the experiment in presence of increasing concentration of double-stranded DNA, as you can see here, to test the selectivity of these ligands for the target. And the most interesting fact is that the unfolding temperature are not affected by the presence of the double-stranded DNA, suggesting that these compounds present discrete selectivity for the target. Then we have evaluated the photoreactivity of our ligands in presence of the biological target. Here I reported the experiments performed on the telomeric quadruplex, but the same experiment was repeated with the other quadruplex sequence. We performed the Sorry, can you hear me?
Yes.
Okay.
Now, yes.
Okay. Sorry, we performed the irradiation experiments with ligands 1, 2, and 4. And because these compounds showed higher reactivity compared to the other tested ligands. So we have irradiated our G quadruplex sequence in presence of the ligands, and we have measured the alkylation yield after at different time of irradiation. And after 8 hours, we obtained the highest alkylation yields with compound 2 and compound 4, the 2 ammonium salts. And this is coherent with the results obtained in the preliminary studies. We performed on the reaction mixture also page analysis to evaluate the formation of the alkylated product. And as you can see here, especially in case of compound 2 and 4, we can see the formation of a new spot, which has been attributed to the alkylated G quadruplex. In the end, to further demonstrate the formation of the adduct with the target, we performed Lcms analysis. Here I reported the result obtained for ligand 2. And as you can see, we observed and we found the mass corresponding to the adduct of the ligand with a telomeric quadruplex. And moreover, we have even observed, even in less extent, the formation of a bis alkylated G quadruplex, evidencing once again that these ligands can act as cross-linking agents and further reinforce the interaction with the target. So in conclusion, we have synthesized a library of novel photoresponsive quadruplex ligands. We have investigated their photochemical behavior. We tested their affinity for different quadruplex structures, and we have evaluated we have demonstrated their ability to alkylate G quadruplex structures upon photoactivation. In conclusion, I want to thank all my research group for the support, in particular my supervisor and all collaborators that contributed to this work. And of course, all of you for the kind attention.
Nice presentation, Alessandra. Any question from the audience? I have some questions for you. The first is, have you evaluated the selectivity against the duplex?
Yes. We have tested the selectivity with a duplex in threat melting analysis, and we have seen that the affinity for the G quadruplex structures isn't affected, is only poorly affected by the presence of double-strand DNA. So the selectivity was quite promising.
Okay, we have a question from Professor Russell. Have you checked the selectivity against other DNA secondary structures?
Okay, no, we didn't test the selectivity towards different secondary structures, so only for our quadruplex structures. Indeed, we have selected the telomeric quadruplex AMYC because in preliminary studies, we have seen that the ligands showed a particularly affinity for this sequence. But no, we didn't test the selectivity with other secondary structures. So it's a nice suggestion. Thank you.
Okay. I have another question. Have you performed some experiments like molecular dynamics or NMR titration to evaluate the interaction with the G4 tetrads?
No, we didn't perform liver computational studies and no NMR studies, but to evaluate the formation of the adduct, it will be really interesting, especially NMR.
Okay, and probably also the expression of oncogenic proteins.
Yeah.
Okay, thank you again.
Thank you. Thank you.
And it's the turn of Dr. Francesco Merlino from University of Naples. Dr. Merlino, are you with us?
Yeah, can you hear me?
Yeah, clearly.
I expected there was a break, but for me it's okay. No coffee break.
Okay. Okay. So he will show us alpha helix inspired stapling of temporin-like peptides, new antimicrobial agents to challenge the antibiotic resistance. So, Dr. Merlino, the stage is yours.
Thank you, Chair. Hi, everyone. Can you hear me and can you see properly my screen? Please,
yes,
can you confirm?
Of course.
Okay.
Yes.
Please, at first, I would like to thank the scientific committee for giving me the chance to present my recent outcomes regarding the development of peptide mimetics of the temporin family endowed with antimicrobial activity. But first, let me spend a few words about antibiotic resistance. This occurs when bacteria change in response to the overuse of the current available antibiotics, and bacteria develop resistance more quickly than usual. And this phenomenon has also a social impact by increasing medical costs and the prolonged hospital stays and increase the mortality. And if you have a look at the discovery of antibiotic timeline, you can see there is a whole about the last 20 years regarding the discovery of new classes of antibiotics with new mechanism of action, and also the current pandemic, let's say, uncertainty is contributing to getting worse prediction about the future. So what to expect? Well, without efficacious antibiotics in therapy, probably we will see a coming back to a pre-antibiotic era. That's why the World Health Organization declared the antimicrobial resistance as one of the top 10 global issues for the public health. And numbers about predictions are really dramatic by 2050 if no action will be taken. And SK pathogens are those of major concerns. They show particular mechanism of resistance and also are accountable for serious infection disease. So in this situation, I would ask how to find an escape to escape. One good thing to do is to look for novel therapeutic approaches, as medicinal chemists have worked with antimicrobial peptides, development of analogs of antimicrobial peptides as the next generation of antibiotics. These are small peptides naturally produced by living organisms as host defense peptides. They possess a specific primary and secondary structures, and but I want to that are accountable of their selected interaction with the bacterial membrane. But I want to show you immediately that they already find applications in several fields, including biomedical application. They were approved, some of them for example, in topical preparations, and the number of those in ongoing clinical trials is increasing as well. My research activity was focused on the temporin family. These peptides were discovered in frogs and present interesting properties that make them good candidates for biological investigations. The mechanism of action is mainly based on the cytoplasmatic membrane perturbation that can happen through one of these mechanisms. And among the 100 temporin isoforms, the temporin L is the most interesting. This is a 13-mer peptide that show the highest antimicrobial potency among temporins, and also as an activity against gram-negative species, and also adopts an alpha helical structure in a membrane environment. But the drawback is the high cytotoxicity at its antimicrobial concentration against human red blood cells. And for this reason, many efforts in the past were aimed at the development of new analogs, and particularly one analog called TL34 was obtained by replacement of Q residues, so local modification on the peptide sequence. This analog showed a less of a helical content in the N-terminal and showed an antimicrobial performance very similar to the TL, but mostly important, it reduced drastically the hemolytic effects at even at high concentration. And for this reason, it was considered the lead compound for the development of new series of peptides and derivatives, and that are reported in several contributions that we have developed in our research group. But we still had a question on our mind about the real correlation between the alpha helical content, especially in the C-terminus region, and the biological behavior, including cytotoxicity for this compound. And so here we go with our medchem story, and about the idea to make new cyclic temporin derivatives, because new cyclic derivatives, an intramolecular cyclization is an approach that in peptide chemistry is used for to improve or modify pharmacodynamic on PK properties. You can choose different side chain cross-linking, like disulfide lactam. The choice depends on many factors. In our design, that again was inspired by the crucial role of the helix content in the C-terminal region of the TL34. We took in consideration 3 objectives in this project. We wanted to identify first those residues that allowed cyclization on the peptide sequence by first considering the lactam than the triazole, because we knew from literature that they can confer more helical content. Then we wanted to define the proper distance between these amino acids in the peptide sequence. And finally, we want to apply different types of cyclization in the same position. And by following this modification, we obtained this peptide library. As for the synthesis, we relied on a solid phase strategy that we have recently combined with the ultrasonication as energy input to accelerate fundamental reaction for the peptide elongation. I'm talking about the coupling that we perform in 5 minutes with the reduced amounts of FMOC-protected amino acids and reagents and coupling reagents, and also about the FMOC removal that we perform in totally 90-second time. The reactor is suspended in an ultrasonic bath in water, in an ultrasonic bath But that's another story. We use the US SPPS method just for the elongation of the peptides. Then during the synthesis, we use specific chemical modifications, chemical reaction for cyclization. Here I just show you the crucial step that is specific for each path. And then the peptides were traditionally cleaved from the resin and purified by HPLC. The antimicrobial activity was evaluated for the lactam and triazole series against a panel of both gram-negative and gram-positive bacteria to define the minimal growth inhibitory concentration. We found two peptides, peptide 12 and peptide 17, that showed they were good antimicrobial candidates. And they actually have the same replacement. I mean, in both cases, the serine in position 6 and the glycine in position 10 were replaced, suggesting us that those can be considered the optimal stapling positions. And in fact, in the second round of the design, we choose the same position and replace the lactam bridge or the triazole with other alpha helix inducer like olefin bridge, then the disulfide. These analogs did not show very nice antimicrobial performance, surprisingly, even for peptide 25. And then a selection of compounds were also tested for cytotoxicity against human keratinocytes. The lactam bridge peptide 12 had low cytotoxicity even at high concentration. Around the antimicrobial concentration, it is really safe after 24 hours of incubation with keratinocytes. And also after 2 hours, the trend is quite the same. And so we were to further investigate about the correlation between activity and alpha helix. And the secondary structures were explored by CD analysis performed in water. And this peptide showed a random coil structure, except for the hydrocarbon bridge peptide 25. While when performed in environment mimicking the bacterial membrane, the alpha helix content increased with different degrees for the analogs, but confirming us that they actually can mimic the alpha helix. They can stabilize the alpha helix. At this point of the project, we want to further investigate the difference between these two analogs, 12 and 17, because of their different performance against some gram-negative strains and some gram-negative bacteria, the difference in terms of potency. So we want to investigate interaction modality with gram-negative bacteria by fluorescence-based assays. First, we look at the same tendency to form helical aggregates at the same concentration, but when we tested them with interaction with LPS, this is a component of the gram-negative bacteria. Well, peptide 17 showed a stronger ability to aggregate at the 20 micromolar with respect to the lactam bridge peptide 12, giving us an explanation why this is less potent than 12. So it seemed that the peptide 12 was the good one, our best candidate, our best compound in our hands. And so we further investigate that by NMR techniques to look at its conformation and also about its ability to induce membrane leakage. And also, and in fact, it was able to make membrane leakage in liposome mimicking gram-positive and better in gram-negative membranes. And also, we tested that for antibiofilm activity and peptide 12 with respect to the native to the reference compound 9, it was able to reduce the biofilm, the viability of preformed biofilm cells at different concentration of 2 strains. And to evaluate the PK impact of our modification, we perform a serostability assay in the presence of 90% fresh human serum. Peptide 12 was really stable, even after more than 6 hours. Actually, the half-life is around 6 hours. That is pretty good for a peptide compound. And so, in conclusion, I hope I convinced you that antimicrobial peptides can be considered effective therapeutic option in antibiotic therapy, and tempering analogs, peptide mimetics, can be a good tool for this aim. And we synthesized the first library of cyclic tempering isotherm, where peptide 12 turned out to be effective for the generation of a new therapeutic agent. Of course, I would like to thank all the people involved in this project, and also I thank you for your kind attention.
Thank you, Dr. Merlino, for your clear and great presentation. And sorry, because I forgot the scheduled coffee break. You tried to say, but I forgot. Sorry.
It's okay.
So, any question from the audience?
Yes. Hi, Francesco. This
Hi,
is
Stefano.
See. So, congratulations for your work.
Thank you.
Looking at your cyclization strategies, I was so curious about some structural elements. For that, I have some questions. The first one, I don't know, maybe I haven't checked properly, but did you resolve the cis and trans, the stereoisomers of your simple peptides? And if it wasn't like that, why? And the second question, if you try to reduce also that double bond in order to not have that diastomers. The third question, then that's all. If you are thinking, considering the good results of the approach to cyclize those peptides, have you considered other cyclization for the future?
You mean for other peptides?
No, for the same sequence. I mean, like clips.
Other types of cyclization.
Yes, correct.
Yeah, sure. I reply first at the third question. Yeah, sure. It's every time it's we offer a piece of the puzzle. So and the library is just a photograph of what can happen on this sequence by cyclization, and sure, you can apply many others. While taking consideration taking consideration obviously the secondary structure, and about this sequence, take care of the amphipathic structure. And I'm replying on your first two questions because in the stable peptides, you mean the hydrocarbon bridge peptide. Actually, the amphipathic, an explanation why it was not really good antimicrobial, but it is very good alpha helix. It can be the void of the amphipathic structure for this peptide. While with lactam the triazole, you still maintain that feature. Hydrocarbon. No, we didn't look at the two isomers. Being an II plus for cyclization, we expect just the trans. That's what the literature suggests us, and even by HPLC, we didn't check more conformation, more isomers. And also these analogs was synthesized by the first author of the article, Dr. Bellavita, and during a a visiting stage in the Grossman lab, and Professor Grossman gave this advice to us. So we really trust in him. The second question, we did not try to reduce the double bond. It can be an option, and I thank you for suggesting.
Okay, Francesco. We have another question from Margherita Brindisi, and she said very informative talk, so she congratulates with you. And she asked if did you ever test some of your compounds in association with other antibiotics?
I thank you, Margherita, for a very nice question, and really good question, because sometimes these peptides works very well in association. We did not try, and the association also can be done also with known antibiotics, and maybe to ameliorate their performance. So, yeah, you mean with other antibiotics? Yeah, that's the good way, and for sure it could be also an option to take again some old antibiotics, and renovate their and potentiate their mechanism. Thank you very much. Yes, nice question.
Okay, thanks, Francesco. And now we will stop for the coffee break, and we will be back at 20 to 4, so 10 minutes of coffee break. So see you later. So we are ready to start. Next speaker is Lidia Ciccone from University of Pisa. She will show us multifunctional compounds inspired by natural derivatives as antioxidant agents and ILR inhibitor.
Can you see my screen?
Yes.
Can you hear me?
Yes.
Okay, perfect.
You can start.
Thanks. First of all, thanks to organize to select my talk. Diabetes is a global metabolic disorder in which the beta cell of pancreatic islet of Langerhans do not produce insulin, resulting in high blood sugar levels. In type 1 diabetes, pancreas fails to produce insulin, while in type 2 diabetes, insulin is produced, but it's unable to bind its receptor. So we can speak about insulin resistance. Diabetes is a chronic disease that stays asymptomatic for a long time. However, high level of glucose can induce several complications. In fact, they can affect, for example, retina, inducing retinopathy, or nerves, inducing neuropathy, or kidneys, inducing nephropathy, and also it can induce several cardiovascular complications, which can be after provoke stroke or diabetic foot, or in general reducing the blood flow. Aldose reductase, also called ALR2, it's a key enzyme of polyol pathway. It's a small cytosolic enzyme which belongs to aldoketoreductase superfamily, and it catalyzes, for example, the conversion of glucose to sorbitol, and the sorbitol after is oxidized to fructose by sorbitol dehydrogenase. Aldose reductase show a low substrate affinity for glucose, and the conversion of glucose to sorbitol through this metabolic pathway is generally non-significant under euglycemic condition. While under hyperglycemic conditions, the polyol pathway becomes operative. So sorbitol is formed more rapidly than it is converted to fructose, and then it accumulates within the cell, inducing osmotic stress. Osmotic stress, together with reducive stress, glycative stress, and oxidative stress, favor diabetic complications. So the inhibition of this enzyme represents a tool to prevent, or at least delay, the progression and the severity of diabetic complications. Starting from middle 60, the scientific interest towards this enzyme and its inhibitors grew up quickly. In fact, several synthetic compounds show very strong ability to inhibit this enzyme, but the problem was that the majority of them failed during clinical trials. Recently, natural compounds have been investigated as a good aldose reductase inhibitors, and this is very interesting because a lot of natural compounds have also a good antioxidant property. So natural compound can be considered as a as a compound with a dual effect, antioxidant and potentially anti-diabetic. The problem of the natural compound is often connected to the low bioavailability. So starting from the good result reported in the literature, we propose a new series of synthetic compound inspired by natural one. And our aim is to obtain synthetic compound with a double activity, but at the same time that are less problem connected to the low bioavailability. So we propose the bisaryllic compound in which two aromatic portions are connected through a heteroatomic linker. And in particular, the ring A was substituted in orthometa and para position with methyl group or chlorine atom, and the ring B was decorated with 2 or 3 hydroxylic compound. So following this procedure, we obtained some new polyhydroxy derivatives. In brief, we obtained the compound 11AE by Mitsunobu reaction between the bromo derivatives 8AE with N-hydroxyphthalamide. And after the protection of thalidomide group in ammonia and methanol, give the desired compounds as hydrochloride salts. These hydrochloride salts were after treated in methanol water in presence of the appropriate aldehyde to give the final compound with a precise geometry. In fact, all the compounds were characterized by E geometry. After the recombinant expression of aldol reductase was done in collaboration with Dr. Carole Frouchard, following her protocol that in fact she developed this protocol in her laboratory. So in brief, the plasmid was designed after it was expressed in E. coli, then the protein was recovered and purified. And the first assay of this production gave a really good yield. In fact, we obtained 140 milligrams of pure protein from 1 liter of culture. Then we use our recombinant protein to test the activity of this compound against the enzyme. And we test all compounds at the concentration of 100 micromolar. And it's interesting that we found 3 compounds, 1B, 2B, and 3B, that show promising inhibitory activity. Then we decide to investigate, to do the first investigation in order to evaluate the potential affinity of this compound with the aldoreductase binding site. It's well known that the binding site of aldoreductase can have a different conformation. In fact, the shape of the binding site change when it's crystallized with different inhibitors. So in this first evaluation, all the 3 compounds were analyzed against the 4 conformation. And it's interesting highlights that we obtain good binding energy for all compounds. The best one was compound 2B, and the best interaction seems to be between compound 2B and the 4JII conformation, PDB structure conformation. After we evaluated all compounds for their antioxidant properties, and the thiobarbituric acid reagent substances assay showed that all compounds tested at 100 micromolar have a good profile as antioxidant. And in particular, it's important that compound which present on ring B, gallic or catecholic or resorcinic groups display higher value compared to the reference compound resveratrol and pterostilbene. And this profile is also confirmed at 10 micromolar concentration. So starting from this result, we decided to evaluate these compounds also for their free radical scavenging using the DPPH assay. And so all compounds were tested at concentration on 100 micromolar. And also in this case, we obtain good value. In fact, they show a percentage of antioxidant activity higher than the reference compounds. And it's interesting to highlight that this profile is also maintained at 10 micromolar. And in particular, three compounds, the compound that present on ring B, the catechol portion, maintain this more or less the same activity. So there is not so much difference between 10 micromolar and 100 micromolar. So in conclusion, in our work, we propose a new synthetic compound inspired by natural one with the aim to obtain a compound with a double activity. And we obtain at the end 3 compounds that show promising profile. Thank you for your attention. Thanks to all the people that participate to this work, and thanks to ProInLab, that is the research group I belong to. Thanks.
Thank you, Lidia, for your presentation. Any questions from the audience? Dr. Coluccia?
So I have just a curiosity, actually 2 curiosities. The first one is about, did you compute the redox potential of the compounds? So often the natural compounds has a redox potential that may affect the results.
No. No.
Just a curiosity. Okay. And the second one was about the docking computation. So it was run docking of the 3 compounds versus all the conformation, all the available conformation of the enzymes, and then you computed the best docking score. I understand well.
At the moment, I present only the energy value because the calculation is still in course. And so our collaborator, in principle, they will furnish the result when the calcul will be finished. Unfortunately, I don't have now.
No, that's fine. It was just a curiosity. Thank you very much.
Thanks.
Okay. I have a curiosity. Have you considered a cyclization of the central linker of your compounds?
No, and we didn't have a the NMR showed that we don't have a cyclization.
No, but for further investigation, you
consider... Okay. No, not yet. At the moment, no. We are we started with we were inspired by Stilben and Calcon structure, so scaffold. So we started from a linear linker. But why not? We can consider also to put a cycle in the middle of the linker.
Also a substitution.
Yes. Yes.
Okay. Thank you again.
Thank you.
And next speaker is Francesco Fiorentino from University Sapienza of Rome.
Yeah, hello. Can you?
Yes, we can hear you.
Can you hear me? Yes. Okay, cool.
Yeah. With a presentation entitled Drug Discovery Meets Native Mass Spectrometry, Unraveling the Modulation of LPS Transport Proteins. So whenever you want, the stage is yours.
So, okay, cool.
Okay.
You can see the screen, I guess.
Yes.
Yeah, so hello, everyone, and thanks for choosing my talk for this conference. What I'm going to talk about today is how I employed native mass spectrometry to investigate on the interactions between molecules, small molecules and peptides, and proteins involved in the transport of lipopolysaccharide. So first of all, I want to introduce you to native mass spectrometry. So this is a biophysical technique that allows the observation of intact proteins in the gas phase. And this can happen thanks to the soft conditions that are used, starting from the ionization of the protein, so from the passage from the solution state to the gas phase, and as well as all the conditions, including voltages and radio frequencies in the mass spectrometer, that allow to keep non-covalent interactions. So this enables us to analyze the stoichiometry of multi-protein complexes, as well as their composition and binding with small molecules. So just to give an idea on how like a mass spectrum, like in this case, it may look like, this is a protein, in this case is a membrane protein of around 35 kilodalton, and the red peaks correspond to the apo form of the protein, while the light blue ones, the adducts here, correspond to the lipid that was co-purified. So it's a non-covalent interaction here. I'm going to give you a couple of examples of the application of native MS to understand the modulation of proteins, one for soluble protein, and another case for membrane proteins. So both of them belong to the lipopolysaccharide transport system. So as you may or may not know, LPS is a glycolipid that is present on the outer leaflet of the outer membrane of gram-negative bacteria. It is very important for bacterial stability as well as protection from the entrance of xenobiotics. This is quite a big molecule. It can range from 5 to 10 kilodalton, and it's quite polar given the presence of a branch polysaccharide. So following its synthesis, which is finalized in the inner membrane, it is transported up to the outer membrane through a multi-protein complex, which can be divided into, let's say, 3 subcomplexes. An inner membrane one, LPT B12GC, which is an ABC transporter, which is the active part of the system, because it couples ATP hydrolysis to the extraction of lipopolysaccharide. Then we have the latest step is LPTDE, this heterodimer that enables the insertion of LPS in the outer leaflet of the outer membrane. And the protein that I'm going to talk about now is LPTA, or LPTH in Pseudomonas aeruginosa. It's an oligomeric soluble protein that acts as a bridge, that bridges the 2 subcomplexes, and is a soluble one in the present in the soluble region, so between the two membranes. What is important here is that this system is a very attractive target for the development of new antimicrobials. However, its structure has only been recently solved for all of the components, so there are not many drugs at the moment. And since LPTA or LPTH bridges the 2 parts of the system, targeting its function, basically going to disrupt protein-protein interaction, can be a winning strategy to develop new drugs. At the moment, only 2 molecules have been described that target the Escherichia coli LPTA. One of them is an antimicrobial peptide, thanatine, which was shown to impair the interactions between A and C, and also D and A. And the other one is this small molecule, this quinoline derivative, IMB-881, which was only shown to impair the interaction between LPTA and LPTC. So also what is important here is that LPTA from E. coli is an oligomer, while in the case of the target of this project, LPTH from Pseudomonas, it was shown to be mainly a dimer in solution, although the equilibrium constant have not been disclosed yet. So what we want to know here is understanding what is the dissociation constant of LPTH, so the monomer dimer equilibrium, understanding whether the molecules that I just mentioned can actually interact with LPTH, since Although the structure is similar to its orthologs, the sequence is quite different. And finally, we were wondering whether we can develop new molecules targeting this protein. To do so, we employed native mass spectrometry, and we developed an assay to assess basically the disruption of the oligomerization of LPTH. This is just the typical workflow that we've been following for this project. So starting from transformation, protein expression, and purification up until the assay using native mass spectrometry. First of all, we had to understand the monomer-dimer equilibrium of LPTH in solution. And what we did was to titrate the protein at different concentrations and quantify basically the monomer and the dimer. As you can see here, the peaks on the left correspond to the monomer, and while the one on the right to the dimer. As we increase the concentration, of the protein, we see more and more dimer in solution. So following the quantification of the molar ratios of the two species, we could obtain the KD, the dissociation constant, which is in the low micromolar. So having established this kind of method to analyze this equilibrium, we then assessed thanatine, which we added basically increasing concentrations of thanatine to 8 micromolar of LPTH. And as you can see here, as we increase the concentration, the dimer basically disappears, while the monomer increases, and in particular, we can see binding between thanatine and the monomeric species. And we could quantify the EC50, so the concentration in which we observe 50% of the maximum effect, which was around in the low micromolar. So this was cool, because now we know also that thanatine can also target the Pseudomonas ortholog of LPTH, so LPTH. We then decided to do something similar with IMB-881. So again, as we increase the concentration of the molecule, we could see a decrease in the dimer concentration, and a quantification gave us EC50 of 4 micromolar, around 4 micromolar, and an Emax. So the maximum effect was 0.73, which means that at the maximum tested concentration, we saw around 73% of monomeric species. So we were also curious about whether we could develop new derivatives of IMB. We initially decided to derivatize differently the nitrogen of the dihydroxadzino moiety, but all the different substitutions didn't give the same good results as IMB, which indicates that probably the phenyl portion here is quite important for its activity. In addition, another problem of IMB-881 is that it is quite insoluble, and even in DMSO we found it difficult to test it. So what would be cool was to develop quinoline derivatives which were more water soluble, and we then therefore decided to test a library of derivatives of this small molecule called IOX1. All of them bear, except for 2J, bear a 4 to 6 carbon acyl chain on the 5-carboxyamide, and may or may not be substituted on the hydroxyl group in position 8. So among them, 4 of them showed EC50 below 20 micromolar. So, and all of them possessed a chain of 4 to 5 carbons, which indicated us that even like 6 carbons, so the aminocaproic acid derivatives were not as good as the other ones. Even like compound 2J bearing terbutyl phenylpropyl carbamate portion here was quite good, but it's obviously an outlier compared to the other, and its log P is relatively high. So if we focus here, we could also draw some initial structure activity relationships, which indicated that the esterification of the carboxylic group was kind of quite favorable for the activity, as well as protection of the hydroxyl with a methoxymethyl portion. I can give you here an example of how the mass spectra look like with compound 2E. So here you can see that this is the protein in the absence of the molecule, while when we added the compound that, like in this case 25 micromolar, we could see a dramatic increase of the monomer concentration. And if now, so from this study here, we could already get some information for the future development of novel derivatives. And what we discovered here is, like, first of all, we managed to establish a native mass spectrometry-based method to assess protein-protein interaction disruptors. And also we got to know that thanatine and IMB also target LPTH. And we got to know that these quinoline derivatives bearing short acyl chains on the 5-carboxyamide also may be good hit compounds for further optimization. So as I said, I also want to give you just some details on application on native mass spectrometry, also on membrane proteins, and I will be very, very quick, a couple of slides. In this case, we applied this on the outer membrane protein LPTDE, which was also shown to be targeted by thanatine. And following purification, we could titrate the substrate, so LPS, and obtain information on its KD again, and also obtain information on the competition. We basically performed a competition experiment, which showed that actually thanatine does not compete with the substrate when interacting with LPTDE. And finally, we also wanted to know whether the protein can selectively recognize the substrate compared to other lipids. When we added an excess of detergent, we could see that while the detergent could compete with lipids like PLPG or cardiolipin, it could not with the substrate, which also gave us some information on the selectivity of binding of LPTDE. This was just to show that another application also not only for soluble, but also for the more challenging ones, the membrane proteins. With this slide now I can conclude, and I want to thank everyone from Sapienza who supported this work, and from the University of Oxford, where I did my PhD and I started some of this work. And I want to thank you everyone for your attention.
Thank you, Francesco, and congratulations for your great presentation.
Thank you.
Any question from the audience?
Yes.
Antonio?
I have a question. Hi, Francesco.
Hello.
Really nice presentation, really brilliant work. Just a curiosity, is the method available to screen a large amount of compounds? So something like high throughput screening, or you need to have a short train set of compound?
I would say for really high throughput, it may be still difficult. We tested like a library of, I showed 20, but like roughly 50 compounds. That is, so it's medium throughput, definitely not high throughput, because so for soluble, for easy proteins, relatively easy ones, like the small soluble ones, it can be kind of automatized also because the spectra are, in particular for the apoprotein, are more easy to acquire. So that in that case, one can develop a high throughput method. For membrane proteins, is still a bit more challenging, I would say.
And the second one, sorry, is another curiosity, is about so the real problem with this kind of protein is to identify a putative binding site.
Yeah,
it's possible to introduce mutation and thus to evaluate the binding. For example, introducing mutation in LPTH and then to evaluate the binding of a possible inhibitor.
Yeah, definitely. Here we monitored more. Maybe you've seen the effect, so the dissociation, and binding. It depends also on the type of molecules that you evaluate. But yes, in principle, it is definitely possible, and we did that for other projects.
Yeah, thank you.
Thank you very much for your question.
Can I do another question? Do you have time?
Yes.
So, Francesco, congratulations for bringing us into the native mass spectrometry. Actually, Antonio scooped one of my questions, but I have some others. I'm so curious about the technique, and I was wondering about two points. The first one, if you can measure binding between protein and nucleic acids with the native mass spectrometry, and the other one is you mentioned that. So the native mass spectrometry, one of the advantage is that you can use not denaturating conditions, but most of the compounds, at least the heat compounds, they do not show good solubility in water. So what happens when you have to cope with this kind of compounds? So the DMSO that you add to dissolve them can denaturate the protein or not?
Okay. So yeah, to answer to your first question, yes, that we can definitely measure the binding, like the interaction between proteins and nucleic acids. There are also studies showing basically applying native mass spectrometry just to nucleic acids. I also personally did that in the past. For your second question, also this is what I did in this project. We compare when, for instance, in the case of IMB. I had to dissolve it in the DMSO, and I did the same. I used the same final DMSO concentration also for the solution that I used for the protein. So even in its apo form. So also the concentration here was relatively lower than the final concentration. I mean, lower than 0.1%. So this, from this point of view, is very similar to normal biophysical assays. So as a control, you basically use the same conditions, solution conditions of when the compound is present. I hope I answer your question.
Yeah. Thank you. Cool. Thank you very much.
Thank you very much.
Okay. Thank you again, Francesco.
Cool. Okay. Cool.
And now it's the turn of our last speaker of our presentation, Sara Battista from University of L'Aquila. She will show us preparation of structurally related liposomes and investigation of their physicochemical properties, antibacterial activity, and synergistic effect with antibiotics. Sara.
Can you hear me? Can you see my screen?
Yes, of course.
Okay, so good afternoon to everybody. And first of all, I would like to thank the scientific committee for giving me the opportunity to show you my results today. And this work will focus on the preparation of structurally related liposomes and the investigation of their physicochemical properties, antibacterial activity, and synergistic effect with antibiotics. In general, liposomes are spherical vesicles that include at least one lipid bilayer and that encapsulate inside them an aqueous core. They are biocompatible and versatile aggregates because it's possible to modulate their properties, functionalizing them and or changing their composition. They are often used as the drug delivery systems because it's possible to encapsulate inside them both hydrophilic and hydrophobic drugs, but they can also exert a pharmacological potential if devoid of an active principle. It's known that charged liposomes show a greater ability to interact with
Sorry, Sarah. Can you hear me? We had some problem with connection. Sarah, we had some problem with connection. So please, if you restart your presentation. Sorry. But we cannot hear you. Probably you have some problem with connection. Switch on your microphone, please. Sarah.
Sarah, maybe you can hear me now. Sorry, I don't know what was happening. Okay, so
now we can hear you. Okay.
Okay, I was talking about liposomal formulation. So I talked about components. I don't know if you heard this part. Okay, so we prepared the formulations through thin film hydration methodology, followed by sonication, and we
It seems that Sarah has some problem with connection. Sarah, we can't hear you. Sarah, are you with us? Okay, Sarah has some problem with connection, so we can skip into the slide and talk. The first is Alice Della Valle from University of Chieti, and she will show us synthesis and characterization of linear peptides analogs of endorphin 2 and methionine, leucine, and cephalin containing a feeding gamma lactam. So, Alicia, please. We cannot see you. We cannot hear you, sorry. Alicia? Turn on your microphone, please. Okay. No. Alicia, you have some problem with the connection, probably.
Alicia, you should see a sign with a microphone at the very bottom, and there is a top-down menu. If we click on that, you should have the option to choose your speaker, and probably the speaker, it's not going with the laptop one, but maybe with headphones or something else. So that's why it's not capturing your voice.
No.
And eventually the last try to disconnect and reconnect to the session, because maybe the setting could be fixed in this way.
Okay. We can skip, and it's the turn of Salvatore Galati from University of Pisa.
Sorry, I am here now. I don't know if I have to wait later. I don't know.
Yes, I will introduce you later.
Okay.
Can I start?
Yes, of course.
Okay. I hope you can see my screen and hear my voice. And good afternoon to everyone.
Yes.
And first, let me say thank you to the organization team for giving me the opportunity to speak in this seminar. Today, I would like to talk about a project that I did at the beginning of this year, and it was based on studying the selectivity of carbonic anhydrase inhibitors. Carbonic anhydrase is an enzyme which catalyzes the interconversion between carbon dioxide and hydrogen carbonate. It's present in different parts of human body, and it's involved in several physiological roles. In particular, I focus on studying the selectivity between isoform 9 and isoform 2 inhibitors. The first one is an isoform related to the diffusion of hypoxic tumors, while the CA2 is the most widely distributed isoform and so responsible for possible side effects of the inhibition therapy. So our first aim was to generate machine learning models that can help on identify new potential selective inhibitors. To do this, we first collected data of compounds which were tested on both isoforms from Campbell database. Thanks to a validated procedure, we obtained at the end 870 selective inhibitors and about 1,600 non-selective inhibitors. In this procedure, we applied many filters, and one of the most important was to select compounds which were tested exclusively in Professor Suporan laboratory in order to work with activity values which were measured in with comparable assay conditions. The next step was to generate machine learning models with an hyperparameter optimization procedure followed by a cross-validation to measure the performance of two models, random forest and support vector machine. As you can see here in the box plot, both models achieved high performance, and so we achieved our first aim. These models can be used to perform virtual screening in order to identify novel potential selective inhibitors. But we didn't stop here. We tried to give an explanation of this high performance of machine learning models. And to do this, we analyzed the features which were important for the model predictions. Among these features, we identified some of them which were present exclusively in selective inhibitors. And with the next analysis, we identified some series of compounds which presented these features. When I talk about series of compounds, I mean molecules which share a common chemical structure. And in the example I reported here in the bottom, you can see a series of compounds which have these selective features, and in particular, they are located in the same region of the molecule. If we assume that the sulfonamide is the head of the molecule, these chemical features are located in the tail of the molecule. A normal question could be, are these features in such way related with the binding mode of these inhibitors? To answer this question, we analyzed the binding mode in the two different isoforms of these compounds. Here, I reported a compound, one of these inhibitors, which was crystallized in both isoforms. It's clear to see that the binding mode that we observe in CA9 and CA2 is different. The cause of this difference is the presence of a non-conserved residue. In particular, it's a phenylalanine in CA2 and a valine in CA9. To have a more clear view, we can observe the animated picture at the bottom of the slide. And you can see how the presence of phenylalanine generates a steric hindrance that doesn't allow the same binding mode in the two different isoforms. We further analyzed this binding mode, and we found that the binding mode of CA9 provides a more stable conformation of the inhibitors. And this can explain why this particular compound is highly selective. Finally, our finding was that when machine learning models assign high prediction score to some chemical features, they are generally involved in structural selective interaction. And in this way, we gave an explanation, we rationalized the black box nature that have machine learning models. I finished my presentation, and thanks.
Thank you, Salvatore. And we could pick up where we left before. Alicia Della Valle. No, we cannot hear you again. Sara Battista?
Yes, I am here. Sorry for the problem, but we have a connection problem here at the university.
Don't worry.
So, trying another time.
And Sarah, just an advice, if while you're presenting, you could switch off your video, that might save a bit of connection for the presentation. So let's try this way.
Okay, I'm trying this way. Okay, maybe I can continue.
Yes, please. The stage is yours.
Okay, so I was talking about the characterization, so the zeta potential, and zeta potential was measured, and we observed that in the presence of cationic surfactants, it was positive and high as expected, and independent from the chain length. On the other hand, we observed that dependence on the chain length in the presence of N-oxide surfactants. For C14 N-oxide, we observed a quite high values, even if the surfactants were deuteraionic. And this result was a little bit expected because we observed it in other works, and can be due to the folding of the pyrrolidinium ring in order to expose to the bulk the two hydrophilic moieties, N-oxide and hydroxyl. And this conformation could be stabilized also by the strong hydrogen bond between these two moieties. And adding two methylenes to the chain length of the surfactant, so the minor component, we observed a decrease of the zeta potential, and this could be ascribed to the chain length mismatch with this component and the DMPC, that is the major component, and that bears to C14 chains. This characteristic in this kind of formulation can influence the thickness, the fluctuation, and surface area, and so the properties of the final aggregates. The calorimetric data were obtained from differential scanning calorimetric measurements, and they give these measurements gives information about the packing and organization of the lipid bilayer. We observed that adding a synthetic surfactant in the bilayer, the van der Waals interaction increased, and as demonstrated by the increase of the temperature of the main transition, the TM, and the decrease of the enthalpy variation associated to this transition. In particular, in the presence of C16 N-oxide surfactant, a broad peak of the main transition and a disappearance of the pre-transition were observed, underlining that in this case the bilayer is less packed, and that the mismatch can influence the organization of the head group area. And these results are supported by zeta potential ones, because in this case we observed the lowest values. All our formulations were tested against a panel of microorganisms, gram-positive, gram-negative, and fungal strains in order to assess the minimum inhibitory concentration, the MIC values. And we didn't observe any interaction between cationic formulations and bacterial strains. This is a surprising result because these formulations were the ones with the highest Zeta potential. That is always considered one of the most important parameters for the interaction with the bacterial environment. And the best results were obtained for gram-positive bacteria strains, and this is an expected result because in this case we don't have the external membrane, plasma membrane, so it's easier to interact. The interaction is favorable. And the best formulations were the one containing antioxide surfactants with the 20%, and this evidence suggests that the presence of antioxide moiety plays a pivotal role in the interaction with the biological environment, more than independently from the Zeta potential of the formulations. And we didn't observe any interaction, neither with gram-negative nor with Candida albicans, probably because in the first case we have the external plasma membrane, so it's more difficult, the interaction between liposomes and bacterial cells. And in the second case, the eukaryote membrane has a different lipidic composition. At this point, we evaluated that the result against the Staphylococcus aureus was very interesting because this strain was resistant to some different kinds of antibiotics, such as clindamycin, erythromycin, gentamicin, and oxacillin. So we decided to test our best liposomes in combination with this antibiotic on Staphylococcus aureus methicillin resistant. And it's important to highlight in order to assess the presence of a synergistic effect. It's important to highlight that the choice of the method used to analyze data could influence the result. Among several models that are available in literature, we chose the FIC model based on the low additivity theory and the Delta E model based on bliss independence theory. The first is more related to the MIC concept, and the second needs only a reduction of the growth to be applied. And in all the cases, we observed a reduction of the antibiotic effective concentration and liposomes too, but in the presence of oxacillin, we observed a synergy with both methodologies, and the antibiotic effective concentration was reduced of 1632 times, falling below the breakpoint values. So the Staphylococcus aureus became sensitive to a drug to which it was resistant. And oxacillin is a beta-lactam that inhibits the synthesis of the peptidoglycan layer of bacterial cell wall. So probably in this case, liposomes can interact with the cell wall and helping the oxacillin to exert its activity. On the other end, with the other antibiotics that needs to penetrate the bilayer, probably liposomes cannot penetrate properly, or maybe they can be disrupted. At this point, we decided to test our best liposomes in combination with oxacillin on other Staphylococcus aureus, in particular 2-coagulase negative Staphylococcus aureus, so Staphylococcus hemolyticus and Staphylococcus epidermidis, and another clinical aureus. And we observed synergy in all the cases and with both methodologies. But in particular, we observed for Staphylococcus epidermidis an antibiotic effective concentration reduction of about 8 times. And another time, the concentration fell below the breakpoint value. So Staphylococcus epidermidis became sensitive to oxacillin. So we can conclude that the results are certainly promising. We prepared and characterized formulations that show the ability to interfere with cell growth, even if they are not properly defined antibiotics. It should be possible to use them in adjuvant therapy, for example, for topical treatment, to improve the activity of antimicrobials such as oxacillin. And the most important advantage is that liposomes don't have a specific target for interacting with cell structures. They are chemotropic agents that are capable to determine morphofunctional alteration of the membranes, causing interference with the function of the cell. So the absence of a specific target is extremely important to avoid the possibility of developing resistance mechanisms with these detergents. And in addition, they are substances with little or no toxicity, so their use in adjuvant therapy will be safe. As a rule, exploiting liposomes property, it will be possible to achieve a dual function, drug carriers and antimicrobial agents. So I would like to thank all the people who helped me in this work. At first, the group in which I work in L'Aquila and my tutor, Professor Luisa Gianzanti, PhD student Elena Allegritti. And then I would like to thank Professor Giuseppe Celenza and its group at the University of L'Aquila for biological tests, and Professor Luciano Galantini from University Sapienza of Rome for dynamic light scattering measurements. At last, I would like to thank again the committee for the opportunity and all of you for your kind attention.
Thank you, Sarah, for your great presentation. Any question from the audience? Probably not. I have just a curiosity. Could the incorporation of antibiotics with the liposome overcome the bacterial resistance mechanism?
We would like to try to encapsulate liposomes in order to encapsulate antibiotics in liposomes in order to assess if there are differences or not with our preliminary tests, because in this study we put the physical together, antibiotics and liposomes, but I don't know the behavior of the system if we try to put the drug inside liposomes. So maybe it is a further investigation. But okay, yeah, probably yes.
Thank you again. And the last speaker of Slide and Talk, Giulia De Soricellis from University of Milan. Balkinase and tryptophan, the keys. Yes, of course. Okay, Balkinase and tryptophan, the keystone for platinum-2 and iridium-3 complexes, to hit the target of biomagic and photodynamic therapy. Please, Giulia.
Okay. Okay, can you see?
Yes, we can see your screen.
Okay, so good afternoon, everybody. Today I will be presenting my project, which is focused on the synthesis and design of platinum and iridium complexes meant to be used as luminescent labels for bioimaging and as photosensitizers for photodynamic therapy. As you can see from the preview, all these platinum and iridium complexes are cyclometallated. They have been widely investigated due to their outstanding photophysical properties. Indeed, first of all, the presence of the heavy metal ion tend to favor the intersystem crossing process, which leads to phosphorescence, and thus, as a consequence, the lifetime of the excited state is very long. Moreover, these complexes can be finely tuned just by introduction of minor modifications on the scaffold to modify the photophysical properties. Also, they show very good stability towards photo and chemical degradation. And lastly, they display a very large Stokes shift, which is very relevant in the context of autoabsorption in order to prevent it. Of course, the main purpose of the project was to enhance the efficiency of this kind of systems, and two approaches were actually adopted. First of all, as you can see from the structure of the platinum complexes that I reported, we decided to insert highly hindered substituents on the paraposition of the pyridine because we hypothesized that the structural configuration of these substituents could be twisted with respect to the molecular plane, thus preventing the aggregation process, which usually leads to loss of excitation energy. This hypothesis was then confirmed by the X-ray analysis, which was performed on the crystalline structure of complex 1E, in which the mesotyl groups were actually twisted of an angle of 61.67 degrees with respect to the molecular plane, which is outlined by the NCN ligand. Under a photophysical point of view, these complexes are outstanding because the analysis performed gave awesome results, in particular in the context of quantum yield, which is around unity for each complex. Talking about the second approach, we focused on the selectivity because when it comes to emitters that are used in the biological environment, it is fundamental to selectively address this kind of emitters inside the specific cells or even inside specific cellular components. Because their action has to be highly localized. So for the purpose, we decided to use tryptophan, not only because it has a central role in the biological environment, but also because it was assessed that the LAT1 transporters, which are responsible for the tryptophan uptake in the biological environment, are actually overexpressed in some cancer lines, for example, in the breast cancer. So we wanted to exploit this kind of phenomenon to enhance the influx of the tryptophan-based emitters inside the cancer cells rather than inside the healthy cells. Here I represented the molecular structure of the three iridium complexes that were synthesized. As you can see, the tryptophan was introduced on the phenanthroline-based ligand, and then three different cyclomethylated ligands were inserted to complete the coordination sphere. The photophysical properties, even in this case, are outstanding because the quantum yield was led to 100% for complex 4, with respect to complex 2, in which the quantum yield was around 50%. The biological assays are still ongoing, and both the platinum and iridium complexes will be inserted inside the brain cells in vitro in order to test their efficacy as luminescent labels for bioimaging and as photosensitizers for photodynamic therapy. And so I thank you for your kind attention.
Thank you, Giulia. And we finished our medicinal chemistry session. So at this time, I will really thank all the speakers, our invited speakers, all the attendances of our meeting. And I think it was a great opportunity for all to share their experience, their ideas, their research. So thank you all for all your contributions, and see you next year in Naples. Bye.